Today's topic is on chaotic dynamics in neurons and neural networks. Now, before we start, let me just give a briefing of chaotic system. What is a chaotic system? If we have the present parameters of a system, you can determine the future. But if there is an approximation or if some minute data is left out, you cannot approximately determine the future. That means future becomes unpredictable, even though it is deterministic. Now, that is a tiny difference in initial conditions is amplified to give enormously different results. You can call this as sickness, that is sensitivity to initial conditions. Now, examples of chaos. There are ample examples in ordinary life. Almost all the physical systems are, I must say, are chaotic systems. The weather system, it is an example of chaotic system where you have even the butterfly tapping may transform the weather or may make a storm in some other region of universe. And that is, we all know that is a butterfly effect. And also we have the heart. Heart is also a chaotic system. Heart never beats again the same way. And in the case of human body itself is a chaotic system. All these are evolving as chaotic systems. That means you cannot predict its course of events, even though you can calculate and determine it using equations. Now, this is an example of sensitivity to initial conditions. This is a system where it starts from two different, slightly closer. They are very close initial points, starting from initial points. Initial points are very close to each other, but you can see the course of the system that is moving away from each other at times, and they are moving towards each other at times. And you cannot predict the system without calculations. That is, they are behaving as a non-linear systems, or they are non-linear systems where their course is very much different from one another. Now, what are the features of chaotic systems? Non-linearity. All these are features: complexity, randomness, unpredictable, but you can find out. Non-integrable. All these are features of chaotic systems. Now, we have the, suppose there are chaos in brain. It's not suppose, there are chaos in brain. And brain, there is ample opportunity, or the brain is behaving as a chaotic system. Since it has more than 50 transmitters, thousands of cell types are there, and complex electromagnetic phenomenology, and there are continuous instability in the case of brain, because it is taking stimulations from outside the brain, and it is getting stimulations from inside the brain. That is, other neurons are also are influencing the neurons. Now, but still, brain is thought of as a chemical point-to-point switch. That means the neurons are communicating between each other, and in that communications, there may be chaotic events. Now, brain function cannot be explained in terms of individual neurons, because the neuron behavior is different from that of individual neurons in the network. And it cannot be explained as a local network, because a large number of neurons are involved in the network, and it cannot be characterized as a reaction to a certain stimuli. Now, in the case of neuron, you can have the biological structure like this. We need not go into the detailed structure of biological structure. And this is the zoomed version of the structure of the neuron. And we are more concentrated on the response of the neuron to each and every stimuli. This is the spike of the neuron. That is, at rest, neuron is having minus 70 millivolt. And as a stimulus comes from outside or from inside the brain, it gives a spikes. And the spike increases up to 30 millivolt. And after the spiking, it will move on to the rest state. And the spiking time is about 2 milliseconds only. Now, the neurons communicate between each other through electrical synapse, that is instantaneous and bidirectional. And it can be through chemical synapse, that is through neurotransmitters. And it is unidirectional and relatively slow. Now, these are different types of spiking of neurons. Neuron can spike as in the case of tonic spiking or tonic bursting, or spike frequency may be like this, or these are different types of spiking that is observed in neurons. There are, you can categorize or divide neurons into three: sensory neuron, motor neuron, and interneuron. Now, the neural network will decide the cognitive process, that is to receive information, transform, store and recover, and put it into work. And it is also dealing with memory and also diseases like seizures, Alzheimer's, etc. Now, all these are decided by the neural network, as we all know. Now, what about brain waves? Brain waves ranges from delta waves to gamma waves. Delta waves are having very low frequency, up to 4 hertz. And that represents actually deep sleep. In deep sleep, frequency is very low. Frequency of the network, neuron network, that is neuron spiking or neuron firing, is very much low. And as it increases to 4 to 8 hertz, you can consider it as a deep meditation or rapid eye movement sleep. That is not deep sleep. And it can be alpha waves, which represents drowsy state. Still, the frequency is higher, 8 to 12 hertz. For beta waves, it represents alert state. The frequency is between 12 to 40 hertz. And if the frequency is greater than 40 hertz, that represents concentration and cognitive process, selective attention. All these are characterized by frequency greater than gamma waves. Now, how many neurons are there in human brain? About 100 billion neurons are there. And in the case of fruit fly, it is only 100,000 neurons. In comparison with human brain, it is very low. It is expected like that. And in the case of mouse, it is still higher, 75 million neurons. And in the case of cats, 250 million neurons. And in the case of chimpanzees, 7 billion neurons. And of course, in the case of elephant, it is greater than that of the human being. It is 257 billion neurons. Now, the number of neurons that we have to deal with is enormously high, 100 billion neurons when we study the human brain. And in addition to that, when two neurons are coupled together, their dynamics become still more complex. After coupling, the behavior itself is changing. Now, because there can be synchronization between neurons, and even the amplitude of oscillation may be, or spike may be coming to flat, or the voltage may not be oscillating, and oscillations that can be expected. And there are also near-death spikes observed in the neural network. Now, when two neurons are coupled together, there may be synchronization. That means one neuron may follow the other neuron. That means some properties may converge to a common value. And that means networked neurons can differ from that of the individual neuron. So not only we have to study the isolated neuron, and the behavior of the neurons in the network must also be studied. Now, the activities of the neurons or neural network depends on the coupling strength, network topology, network size, type of neurons and type of networks, noise, and electric fields. All these are to be considered when we consider the neural network. Now, the theoreticians model the neurons. There are different models which characterize the biological neurons or which mimic the biological neurons. And some neuron models, all these neuron models are studied by finding out their evolution or the evolution of the neurons through differential equations. Now, some neuron models are easier to compute. At the same time, they mimic the biological neuron. In some cases, two equations are there, and it is easier to compute. Computational time will be less. In that aspect, Ishkevich model that is proposed by the scientist Ishkevich, and that gives more or less the same biological behavior of the neuron, at the same time easier to compute, and computational time is less. Now, in this work that is presented here, we will give a brief discussion of the neural network of C. elegans. C. elegans is a soil worm. It feeds on bacteria, and its size is only about 1 millimeter, and it has only 302 neurons in it. And most of the C. elegans are having both male and female organs, and few of them are males. And they are having it is a multicellular organism where the complete connectome or neural path is identified. That is, connection between neurons, all the connections are identified by the scientists. So we will move on to the study of C. elegans. Why C. elegans is important? Because 35% of its genome is similar to that of humans. That means 1/3 of its genetic code is same as that of the humans. So it can be used to understand human brain. That is why we have chosen C. elegans as our study material. Our neural network is chosen or is that of C. elegans. And this C. elegans is having both chemical synapses and gap junctions. Gap junctions means electrical synapse. They communicate, the neurons communicate among themselves through chemical synapses, that is neurotransmitters, and also through the by the electrical synapse. Now, a synapse is a structure which permits a neuron to communicate among themselves. It can be electrical or chemical. Now, it has a modular arrangement. The nervous system has a modular arrangement, which is the same as that, similar to that of human being. Now, we will examine here the contributions of individual neurons towards the behavior of the network and influence of chaotic neurons. Suppose neurons are made into chaotic. What happens to the system? That is our concern. Now, these are the two equations that that is used to represent the neuron, and that is given by Ishkevich. This W represents the action potential, that is membrane potential of the neuron. K gives the current. As we have already stated, as the voltage rises up to 30 millivolt, it will be resetting to a value, and that value is chosen as C. Or from 30 millivolt, it will reduce its potential and it will be subsided to minus 70 millivolt afterwards when it comes to rest. And this is the reset value for the current. And these are the different parameters which are given to the equations. And these parameters will decide whether the neuron is behaving like a chaotic neuron or not. Now, we have already seen there is a current term, and that current term is supplied by, can be from external, or it will be also supplemented, or it will be also added by the adjacent neurons. And this G synchronization will give you the coupling strength between the neurons. And AJI is the connection that will give you the connections between the neurons. Jth neuron will be connected to the ith neuron. So the influence of ith neuron is examined by or given by this AJI. Now this is the connection of local network of 131 frontal neurons. There are only 131 frontal neurons out of 302 neurons. 131 are frontal neurons, and they are connected like this. This is already given, or it is already investigated earlier, and it is given in Bermatless. Now this adjacency matrix gives help us to find out the connection between the other neurons. Now we will examine here the firing rate and the synchronization and the entropy of the neural network. And we will then we will find out the network behavior. We can choose parameter values to A, B, C, D values that we have already seen in the equation. And you can make the neurons by adjusting these values to that of chaotic neurons. You can simulate the individual neuron behavior dynamics and find out that whether it is chaotic or not. Now, when we have started from the coupling constant is 0, that means all neurons are independent of each other. There is no connection between them. In that case, these are the firings of various neurons. There are 131 neurons. At different times, they fire like this. Each firing is given by a dot. And when the coupling constant is increased to 2.67, the network firing has become minimum. That means if the coupling constant is increased, or if the connection between, if there is more influence from other neurons, the firing rate is reduced. And then if we increase the coupling constant to 10, the firing rate again increases. Now, what happens in the case of chaotic neurons? In the case of chaotic neurons, now in the network, we have introduced there are total of 131 neurons. We made 60 of them chaotic randomly. And we have studied the firing rate or the firing of each one. See that some of the neurons have stopped firing after some time. Only a few neurons continue to firing up to 3000 milliseconds. Now, after 56 milliseconds, some of them have stopped firing. And more interesting is that when we made all the neurons as chaotic, all the neurons are made chaotic, and in that case, all of them stopped firing after certain time. At around 600 milliseconds, all of them stopped firing. And we when we expand this figure, we can see that before they stop their firing, you can have a continuous surge of firings, that is potentials. Potential is coming one after another just before, that is the potential is given by the neuron, or they spike continuously just before they die out or they stop their firing. And you can call that as near-death surges. Near-death surges are sudden increase in activity in the human brain before the neurons end their firings. And that is an interesting phenomena which is not predicted earlier theoretically, even though it is observed in 2009 in EEG. When you examine the electroencephalogram, this near-death occurrence phenomenon is observed. Near-death surges are observed before the neuron ends its firing. Now, the reason for this behavior, scientists have identified that it is due to lack of oxygen in brain. That is usually observed in COVID patients also. That is a state of hypoxemia. Due to this hypoxemia, the sodium potassium potential is lost. and hence as electrical activity is rising fast and there is a subsequent surge in electrical activity, and after that the neuron goes to death. And this EEG near-death experiences are observed in 2009 by the scientist Chawla, and that is a supporting evidence for the theoretical conclusion or theoretical prediction. Now let us examine the firing rate. When you examine the firing rate, we have plotted the coupling constant or the connection between the neurons is strengthened one by one and it is increased up to 10. And the number of chaotic neurons in the network, total there are 131 networks, 131 neurons, and out of which we make the neurons chaotic step by step. And we will examine the average firing rate. Now, these different colors will be representing different frequencies. If it is blue, that corresponds to 7 to 9 hertz frequency. And when it comes to green, it is 11 hertz. And when it is red, it is 50. Now, when the number of chaotic neurons is less, then you have higher frequency. That means red color is possible, or red, the frequency corresponding to red, this red region is possible when the coupling constant is between 8 to 10, or there is bonding, more stronger bonding between the neurons. That means it represents higher frequency, means which represents beta waves. Beta waves are representing conscious state or logical thinking, memory activation, alertness. All these are represented by this red region. And that is present when the number of chaotic neurons are very few. At the same time, the coupling between the neurons is stronger. And when you introduce more and more chaotic neurons in the network, what happens is the firing rate reduces to that of blue region, that means 7 hertz, which corresponds to alpha waves. Alpha waves represents a drowsy or relaxed state, and it is having lower frequency. That means if the neural network is having higher number of neurons, that can be that is subsiding the neural activity or neural firing, and the brain goes to a drowsy or relaxed state. And when all the neurons are, when the neurons are made chaotic, when more neurons are made chaotic, there is more synchronization between the neurons. That is, they fire alike if the number of chaotic neurons are increased to 131, or it is maximum is 131. That is also an interesting phenomenon to be noted. And what about the entropy? Entropy also increases with the chaotic neuron number, and also it increases, it also depends on the coupling constant. As the coupling constant is increased, the entropy increases. Now, what we have observed is more chaotic neurons in the network change the network to a resting state. That is, alpha waves dominate. Less chaotic neurons means it is representing alert state, or beta waves dominate. 12 to 30 hertz is the firing frequency. Logical, it represents logical thinking, memory activation, etc. Now, what we understand is, to soothe an overactive brain, some of the neurons can be made chaotic so that the firing rate can be reduced. And also, when the chaotic neurons are increased in the network, it displayed near-death like surges. That means if you can see the neurons are dying after some time, and before they die, a near-death surge is produced by the neuron. Now, more chaotic neurons means network activity is independent of coupling strength. We have seen for the higher chaotic number of high chaotic neurons, it is not depending on the coupling between the two neurons. And also, if there is only less number of chaotic neurons, that means there is strong dependence on coupling strength for the synchronization, firing, and entropy. All these increases with the coupling strength. And near-death experience is not reported earlier by the theoretical methods, and that is reported by this work. And further experimental studies to be performed before its use in treatments like stress, over-anxiety, ADHD, Alzheimer's disease, Parkinson's disease, seizures, etc. Now, with this in background, we can also examine the presence of LIPI noise or influence of LIPI noise in this network. We have to examine what is the influence of LIPI noise in the neural network. Noise can be from inside the neural network or from outside. External or internal electric and magnetic fields on the neural environment can act as a noise. Noise can suppress neuronal activity and cell growth. And also noise can induce synchronization and resonance. Now we will, in this further, we will examine what are the influence of LIPI noise on ceasing or enhancing the wave of death activity, and what are the influence of the LIPI noise on the spatiotemporal activities of C. elegans. LIPI noise is characterized by small fluctuations. It can be random, small and large jumps, and it can be also associated with heavy tail, and that depends on different parameters of the LIPI noise. LIPI noise model the noisy environment of neuronal network. The corresponding to this is an example of LIPI noise influence on a process. Due to this LIPI noise, a process may evolve like this. And this is another example. Due to this LIPI noise, a process can evolve like this. That is, the evolving of the system becomes chaotic, or it is not regular. Now, LIPI noise is introduced in the current. LIPI noise is attributed, or it is added to the current. LIPI noise is characterized by certain parameters like alpha, beta, capital D. D is giving the intensity of the noise. Beta will give the symmetry of the noise. Alpha will give you the stability parameter. If alpha is very high, that is the maximum value is 2, it will be tending to Gaussian noise. Some of us might have heard about Gaussian noise. When alpha is 0.5, you can call that it is characteristic of the LIPI noise, typical of LIPI noise. And we will examine the influence of the stability parameter and scale parameter of noise and coupling constant G synchronization on the network. These are some observations. This is also plotted in the same way as in the last slides. That is, alpha is made to be equal to 0.5. You can see that firing rate is given along with this colors, and the variation with respect to D and coupling constant is studied. D represents, or D is representing the intensity of the noise. Desynchronization is representing the strength of connection between the neurons. Now at alpha equal to 0.5, the first diagram is for alpha equal to 0.5. In that case, see that the region is from 12 to more or less red or dark color, 12 to 18 hertz, which represents beta waves. And the right one, that is, this is the second one, which is plotted for alpha equal to 0.8. In that case, the frequency rises up to the yellow region. The yellow region represents the 40 hertz in this case. 40 hertz means gamma waves. Gamma waves means that is more cognitive concentration and selective concentration, cognitive process are made available when this noise scaling parameter is increased to, stability parameter is increased to 0.8. Now, if all the neurons are made chaotic, alpha equal to 0.5 means the firing frequency is subsided to that of 4 to 8 hertz. That means the neural network will go to sleep, or it will be a meditation state, a somewhat rather sleepy state. Now we have examined the variation of firing rate with respect to alpha and coupling constant. Now see that at alpha equal to around 0.8, the frequency rises up to 40 hertz. That represents gamma waves. Gamma waves will be representing, as we have already stated, concentration and cognitive process, very selective concentration. All these are represented by gamma waves. But still there are delta waves also, which are represented by this dark region, black region, where the frequency is very small and the brain will be in the deep sleep state. That means when the noise stability factor alpha is decreased to 0.2, below 0.2, the brain goes to deep sleep state. All these are interesting phenomena that are observed for variations for LIPI noise. And when all the neurons are made into chaotic neurons, the previous one was non-chaotic neurons, and this is for chaotic neurons. In this case, the frequency rises only up to theta waves. Theta waves means again sleep state or meditation state, or the frequency which is corresponding to gamma waves is not available if all the neurons are chaotic neurons. If the neurons are turned into chaotic, that means there is no possibility or scope for high concentration or cognitive processes. And we have, this is for non-chaotic region, synchronization is studied. And the synchronization, higher synchronization is observed for less value of alpha. As alpha is increased, the synchronization gets reduced. And if the neurons are made into chaotic, then the synchronization activity is lowered. That is, the response of neuron alike with the other neuron is reduced, or synchronization is reduced if the neurons are chaotic. This is again the near-death spikes, which is observed in the case of neurons. And the neurons stops firing after about 500 milliseconds. See that it is, the scale is 0.5 milliseconds, that is why 500 milliseconds. After 5 milliseconds, the neurons goes to rest state. And this is the case of interneuron. We can select an interneuron, and this interneuron will be going to cease firing at t equal to 100. That means interneurons and other neurons respond to the response to this spiking in different manner. As the time goes, the interneuron stops firing very fast. And this is the case of, here in this case, the network is displaying this synchronization for a period of first to diagram for a period of 64 milliseconds. Then this network stops firing. And this is the case of non-chaotic neurons we are discussing. And the first diagram is for alpha is equal to 0.2. And if alpha is increased to 0.7, Then the neurons continued to spike till about 3000 milliseconds. And after that, and only some of the neurons exhibit near-death spikes. That means the noise influences the neural network, and the noise stability factor is an important factor which influences the neuronal behavior. And when the alpha is further increased to 1.5, there is asynchronous bursting. All these are plotted for non-chaotic neurons. And when we examine the total current with respect to alpha, variation of total current with respect to the stability factor, you can see that when alpha becomes 1, the total input current becomes steady, or there is no fluctuation at all. Fluctuations are existing between about 0.3 to 0.7. And average feedback current, that is from the current from other neurons. There are wild fluctuations as alpha is increased to 2. The maximum value of alpha is 2. Initially, there are no such fluctuations, but when it is increased to 2, average feedback current increases. Now, with the discussions, what we have examined here and the results are at alpha equal to 0.7. The firing rate peaks around at alpha equal to 0.7 and then decreases. It has become maximum, and the gamma ray waves are resulted in at alpha equal to 0.7. And near-death surges are also exhibited by this network in the presence of LIPI noise. Even the non-chaotic neurons are present in the network. In the earlier case, in the absence of LIPI noise, we could observe the near-death surges only in the case of chaotic neurons. But in the presence of LIPI noise, even the non-chaotic network or chaotic neurons displayed near-death surges. When alpha is less than 0.8, bursting synchronization and near-death surges are exhibited. At 0.8, gamma oscillations, that is very high frequencies exhibited that we have already stated. And when alpha is further increased, asynchronous firing is exhibited. Now, chaotic neurons reduce the firing rate from beta to delta. That means from alert state to the deep sleep state. The network went to deep sleep state when the chaotic neurons are introduced. And near-death surges are also detected when the chaotic neurons are present in the network. Now, some of the results of this work support transition states of different degrees of synchronization which are observed in the cortical region of the brain of rats. That is experimental study, which is reported in these works or in these journals. And these two published works are given in Nonlinear Dynamics, which is published by Springer. Now, what are the future projections? Future projections you can extend. We have considered only the 131 neurons of frontal neurons of the C. elegans. We can extend that work to 302 neurons and compare with the experimental observations. And you can find out the experimental applications of LIPI noise and chaos to decrease or increase the neural activity, and the role of chaos and noise in controlling diseases like Parkinson's diseases can be examined. With that, I conclude my talk. Once again, thank you for the organizers and the distinguished audience. Thank you for the patient listening.
Thank you, Dr. Rose Ignatius. Honestly, it's been the fruit of a very rigorous research, and that's highly appreciated. Hearty congratulations to you. I have been taking us to the mysterious world of neurons and neural networks, started with the very fundamental notions, then eventually progressed into the technical dimensions, So in a nutshell, we understand that it's so much connected to our life, the moods, and even the technical terminologies like neural network or neuron firing, near-death surge, and the Levy noise, how that can be applicable to controlling diseases. So very interesting topics, and I'm sure there are many areas to be discussed, and you have also projected the future prospects. So wish you all the best with your future research projects. Now we have a session of interaction. Actually, one of the unique features of our LIPI Science Talks is to an opportunity to interact with the resource person. And I'm glad that many scholars are present here and students are also there, researchers there. Dr. K. Babu Joseph, Dr. Jacob Thomas, Dr. Kuruvilla Pandikata, Dr. Nina Joseph, Dr. Sister Bina Jose, Dr. Jansi, and many scholars, researchers are there. Then students who are interested in the topic are also present. Over to the floor for any question. Feel free. This is absolutely feel at home. We encourage everyone to interact one another. Need not be with the resource person as such alone. Yeah. Maybe I would request Dr. K. Babu Joseph, the principal advisor, and who happened to be the guide of our resource person today. And maybe may I request Dr. K. Babu Joseph to begin with your response.
Talk today, it is clear that nonlinear dynamics is more in tune with our day-to-day life. The topic which he has enlightened us about is the brain. I have a couple of questions, specific questions to ask you. You have selected only one particular model, the Kiewicz model, because of the simplicity you said. Because there are only two equations, two first-order coupled equations. The other cases, there may be more equations. Has anybody made similar analysis in the case of other models? That is my first question. Second question is, how do you arrive at these models? For example, the model which you have described, this Kiewicz model, especially the choice of parameters and so on. The study of Lawrence, in the study of chaos, historically, the certain models were tried. The Lawrence model is the best example, but it was derived by truncating certain nonlinear equations of fluid dynamics by approximating those equations. But I fail to understand how one can arrive at phenomenologically looking equations like this. They seem to come out from the blue without any prompting. Do you know how these equations were discovered?
Now, in this case, when we describe a neuron, we must find out the course of action potential or the action potential, how does it behave, and also what happens to the current in the neuron. Now, current and action, the potential of the neuron can be measured experimentally, and the evolution or the states can be found out experimentally. I think these scientists have proposed this model to match with the biological observations. That is, Kiewicz model is true. Kiewicz model is having only two equations. In the case of HH model, there are more number of equations, and it is very difficult to compute to have the computational time. And Morris-Lecar model is also shown in the slide. That is also a similar model. And the models, scientists formulate this model to give the course of action potentials and current. And whether they are becoming chaotic or not, they have randomly changed the parameters and found out the chaotic regions of this evolution of these models.
Another question. You mentioned about the possible applications in neuroscience. Have you interacted with medical doctors?
No, they are reporting from experimental side, that is, I have already shown. In the case of rats, experiments are going on. In human being, this experiments are not yet done. That is a future perspective, and that is to be implemented. Before that, it is to be tried in smaller animals, and after that only it can be I think it can be experimented in human brain.
I think that neuroscience scientists do experiments on smaller animals rather than on humans.
Yes, sir.
So there is a possibility of collaborating with them. So I suggest that you contact scientists at the NEURONS, Bangalore.
I will try. I am trying that. I am trying to contact with the doctors in various hospitals so that they can support with this theoretical studies.
Yeah. Wish you all the best.
Thank you, sir.
Thank you, Dr. Babu Joseph. And the floor is open. Anybody, any area of your interest, something connected with the topic of our presentation, you are welcome. I see a good number of comments in the message box congratulating you, Dr. Rose. Dr. Nina Joseph.
Hi, ma'am.
Thank you.
Good evening. Thank you so much for the wonderful presentation. One question I would like to ask you is regarding this. I've heard that the neurons which are wired together fire together.
Yes.
Yeah. So has it got any connection with our behavior patterns? And if so, how it can be implemented positively to change the behavior?
Surely, it will be affecting our behavior or cognitive processes and our ability to understand, retrieve information, and act accordingly. That will be influenced by this synchronization, that is coupling between the neurons. And how we can influence, that is not the influencing with this Levy noise and all, that is in the primitive experimental stage, that is tried in rats and all, and I think in near future it can be tried to human beings also.
Okay, and one question is with respect to the when you get into the meditative stage, which you were mentioning about that, no? That is alpha.
Yes, meditative state means delta waves.
Delta waves.
Sorry, deep sleep is delta waves, and theta waves is a sleep, that is random eye movement sleep, and alpha waves are drowsy state. Yes, meditation.
Okay. So it is possible to we can move into that state with our own
that is our own intention. And that is as we are tired, I think all these are happening. As we are tired, we are normally moving to the deep sleep state or drowsy state. And that means the neurons may be changing its character. It may be changing to this chaotic nature. And that means it may be, firing is reduced, firing rate is reduced and changed to drowsy state or meditation state.
And you were
automatically it is coming like that. An external intervention that is to be investigated.
Okay, that near death this thing you were mentioning, like I was thinking like there is something called a catatonic state wherein a person moves into a catatonic state and he is not able to do any kind of activities. So has it got any connection with this particular near-death experiment, that near-death stage of neurons, wherein after that stage he is unable to do anything?
No, after that, after some time he can retrieve the activities. But in the case of neurons, when it is died, it is died.
That's permanent.
Yes, but number of neurons in the human brain is enormous. Almost very few neurons are active at a time, and we can make more and more neurons active. Even if when the temperature increases by 1 degree, as we have fever, when the temperature increases by 1 degree Celsius, enormous number of neurons go to death. But more and more neurons are there which are inactive. They are made active, even though some of the neurons are died off.
Okay. Jason, could you introduce yourself briefly, since you asked many questions? We would like to know.
I'm right now sitting in Bangalore.
Okay.
My office. So we are into a project here.
Okay. Thank you.
as in, thank you.
Over to Nina Joseph. Thank you,
Yeah.
Jason.
Yeah, thank you.
Thank you, Dr. Rose. I could not follow, to be honest, I could not follow many of the things which you were telling because my background is different, but it was very intriguing and actually it was mesmerizing, to be frank. And then I was having a doubt, when a person is under the influence of alcohol or drugs, what will happen to this firing? The chaos might increase, I don't know. Can you please enlighten me on that? And again, when it is a more complex human brain, complex brain of advanced animals, the number of firings and the number of interconnections will have a phenomenal increase. So to analyze that, do you think that artificial intelligence, once the brain is connected to some devices where all this can be traced, do you think that application of artificial intelligence will provide a quantum jump to this? And again, yeah, these are the two questions which I have in mind. Thank you.
In the case of neurons, regarding the drugs and alcohol, again, the neurons communicate with each other by neuron transmitters. That means chemicals. Or I think these chemicals will be influenced by this external drugs and alcohol. And due to that, the firing rate may be changed. That is why the person which is having more higher, that is enormous quantity of alcohol, is going to drowsy state, or even if he goes to deep sleep state. And that may be due to the influence of this external drugs and alcohol on the neurotransmitters. Neurotransmitters is also a way to communicate or between the neurons in the neural network. And regarding the artificial intelligence, yes, of course, artificial intelligence may be enabling. That is, it may be, we can introduce LIPI noise, and the neurons can be converted into chaotic state by means of certain simulations or certain computerized mechanism. And due to that, it can be controlled. It can be changed. I think so.
Okay, thank you. And is there any relationship with intuition? When your intuitive powers are stimulated, does it have some connections with the neuron and the transmissions between among them?
Intuitions means we are having some perceptions. It may be influencing. That is communication between the neurons without any cause, external stimuli. There may be communications between the neurons due to their change in currents, change in voltages, and that may be automatically coming due to the presence of certain neuron transmitters. All these that is illusions and false illusions are due to different intensity of different concentration of neurotransmitters. Certain chemicals or medicines are given to improve the neurotransmitters or to increase the neurotransmitters amount, so that this false illusions are reduced in brain.
Okay, thank you so much.
The next, anyone from the floor? I see Kuribula, Dr. Jacob Thomas, and many others there. You're welcome.
Dr. Rose, thank you. Can I be heard?
Yes,
Yes.
Dr. Rose, thank you very much for the wonderful perspective and the very many new insights that you have gained. I'm very sure in the next 10 to 15 years, you will be able to give so much of information, so much of knowledge about our inner self, our consciousness, and how all this brain will function. That will definitely change the way science and the way society will deal with ourselves. So it was a wonderful talk. Now I have two small questions. One is, in your laboratory experiment, how do you transform the normal neurons to chaotic neurons? That is one technical question. And the second question is, this whole question of, near-death spike. To some extent, it can be seen even in the human beings, towards the end, when we are approaching the end, not only in the near-death experiences, but in normal things also, there is that last urge to search and then it collapses. Do you have such explanation why this near-death spikes occur and how it is applicable to our day-to-day human life?
The first question, that is neurons, to mimic the neuron activity, you can surely you can that is you can realize it in laboratory by means of getting electrical circuits which obeys with these equations. These equations, that is electrical circuits can be constructed which obeys these equations and evolve accordingly. That is artificial neurons, the neurons which can mimic the actual neurons with these equations, with the behavior of these equations, can be constructed in the laboratory. And regarding the next question, we have also seen before death there is a surge of activity from the patient, and after that unexpectedly the death comes. And that is also observed theoretically in the case of neurons. The neurons before just if they are chaotic in nature, if they are converted into chaotic nature, they will be giving this near-death surges. Now, converting neurons into chaotic in the brain, that is not physically done now, but I think the neurons are converted into chaotic nature as some neurotransmitters and electrical activities are introduced in the neuron due to various reasons, maybe disease or some other external stimuli. The neurons are converted into chaotic nature, and due to that chaotic nature, they are exhibiting near-death surges, and after that, neurons go to death. Now, the neurons are the last ones which go to death. Before all other neurons will go to death first, and only at the last, the neurons will be going to death. That is an exception to brain death in the accidents. Normally, all the neurons go to death. All the organs go to death before the neurons die. Is it okay?
Does that give any explanation for the out-of-body experience, which people just escaping death are narrating? I don't know whether it's an appropriate scientific question, but just curious.
That means during that near-death surge, that is increase in electrical activity, if there is some intervention due to some medicine or due to some external stimuli, if that stopping of firing of neurons can be prevented, and then the neuron may continue its firing, and that must be what is happening in the actual life. That is due to some intervention, due to some medicine or external stimuli, the neurons may be prevented from exhibiting this near-death surge, and also from stopping firing.
Yeah, there is a question from George Anthony. George, would you like to ask? If you would like to ask, you can just unmute and ask.
Yeah. So I think the answer was to some extent given by Rose. It actually I think comes up. My question was, I'll just read the question. I wanted to know what is the physical equivalent of this near-death experience of this neurons. I think Dr. Jose managed to reply that even before I asked the question. But if you have anything more to add, it will be nice to know.
That is already shown in the case of electroencephalogram. This near-death surges are observed. That is observed in 2009 experimentally when we examine the EEG of the brains of patients, and before they go to death or they go to die, before they die, we can find out the increase in surge of electrical activities, that is firings. The EEG shows an increase in potential, or the number of firings, oscillations increased before they go to death. That is the experimental observation that is done so far. When we examine the EEG that is observed in 2009, there is a surge of waves before the patient dies.
Yeah, that I understood clinically. It could be evaluated due to some readings of or electrical signals, but will actually a person experience those surges physically too? Not only in the neurons, but physically the person will he become more active, more violent, or something like that?
I don't know exactly, but we have seen some patients before they die, some surge in activities or increase in activities. They just we have heard that some patients before they go to death, that is they show some improvement in activities, that is they show some physical increase in physical activities, and after that it is subsided and the death happens. It may be due to this increase in neural surge, neural activity surge.
Thank you. Thank you.
George Anthony, what's your designation? Where are you from? Just out of.
Interestingly, I'm a classmate of Rose.
Oh, come on.
We did our MSc together, and right now I'm the registrar at TIFR, Tata Institute of Fundamental Research.
Oh, wonderful joining us. Thank you so much for joining us. Wow, good.
It's a privilege. It's a privilege.
You're welcome. Anybody else? Okay, hello, George. Anybody else? We are towards the end of our discussions, but one or two more questions would be appreciated. Dr. Rose, I have a question that is something, a humorous question. That is something in line with Dr. Nina Joseph's question. Nina asked, the size of the brain. When you presented the statistics, you said human person's brain is of 100 billion. The number of neurons in the human person's brain is 100 billion, whereas that of an elephant is 257 billion. And he said that, the capacity of the brain or the sharpness of brain increases according to the complexity of the brain. So my question, what way an elephant is superior to human from a neuroscientific perspective, apart from the size of the brain? It has potentials, isn't it? It's so big, not the physical strength, but intellectual or mind level or brain level. I thought of this, that's all.
Regarding the intellectual level, it may be true. That is, the human being is having highly intellectual compared to that of elephant. But the brain has to control all the activities, all the parts of the body. And in order to control all the parts of the body, I think elephant needs a higher number of neurons because it is having larger size. That may be the reason.
Right. Okay, this is okay. It's on different aspects of the functions are taken into account. Thank you.
Yes.
Thank you for
Can I continue with that? Because female brain and male brain. Male brains is everything may be proportional to the human body size. Males have an average human body size greater than that of female. So elephant is inferior to human beings in the brain activity. So does brain size has anything to do with intelligence? What about the density of the neurons? Is it different in males and females? Or is the number of neurons that matters, or is the number of interconnectivity between the neurons which matter more?
That means in the case of male and female, I think intellectual level is the same. But regarding the size, the male is having a little bit larger size, but the number of neurons which are extra needed is only very few, because we are talking in terms of billions. Even if 100 billions are added, then also we will say it is of the order of 100 billion. Even if 1 billion is added, we will say that it is of the order of 100 billions. I don't know whether this male human brain is having a slightly higher number of neurons. It also depends on the networking and the connections and coupling strength. All these are also influencing the activity. But now, regarding the number, I don't know whether it is slightly higher in the case of male brain that is having larger size persons.
Even among males, there may be variation in the number of neurons,
Yes.
the order of a few millions.
Yes.
So the answer that Dr. Rose has given is the most plausible. That means there is no significant
difference
between male and female.
female.
There may be only superficial differences due to the number variation.
Yes, sir.
Okay, maybe the final question, if at all there is somebody, you are welcome.
I have a final question.
Okay.
It seems there is a school which claims that the self, the idea of the self, emerges from the complex behavior of the brain. Self means I-ness, the feeling of I being I. Is it possible to model the self using this approach? Not necessarily the two coupled equations. We may need many more equations of this type. But has anybody, are you aware of any contributions made by anyone, anywhere, towards understanding the self?
To describe ourselves, that is oneness.
Consciousness.
Okay.
Yeah.
Now, I don't think so, because in the case of human brain itself, since the number of neurons are the number is very high, it is not yet investigated, because the computational time will be very large. Even 131 neurons we have investigated with 1 or 2 weeks. The coupling is done, and the evolution is examined with 2 weeks with the 4 parallel processes together. And that means a human brain, in the case of human brain, since the number is very large, I don't think that the investigations are done with the human brain network, because the number is very high.
I don't think, I cannot understand why there is such a variation in the quantity of energy needed for the brain and the quantity of energy needed for a computer to do similar computation. So there is a large scale difference between the two.
And we have to examine the connections between all the neurons, and in order to introduce the connections and coupling parameters, more computational time is needed.
But the brain does the computation with very little energy.
That's
millivolt, millivolt.
Yes, sir. A human brain is able to do that, but with the computer, we found difficulty. Yes.
Yeah.
So
amazing.
Are there some popular books on this topic for a lay person, not for the scientific fraternity and community?
Yes, I can suggest
very simple terms, like Neurons for Dummies or something like that.
Popular books, I don't know for the time being. I can find out and let you know if you give your phone number.
yeah, Dr. Nina is associated with our institute, so she will be connected.
Yes.
Thank you, Dr. Ross. I see Surya Gayatri there and Ramlet. If any final comments, otherwise we finish. Okay.
Dr. Ross, do you have any idea about the neuroplasticity?
Neuroplasticity means you mean that neurons are replaced by some other neurons.
Production of neurons.
Oh.
That is neuroplasticity.
That is artificially. That is artificial neurons. Okay.
Naturally, there are even at the old age there is a natural reproduction of neurons. There are evidences, biological evidences, experimental evidences.
I don't know much about that, but I know that even if some of the neurons are destroyed, other neurons can become active. Since 100 billion neurons are there, only a few are active, and when some of them die, the other neurons are become active. And yes, I don't know about this that is neuroplasticity.
Yes. Okay.
Thank you.
Thank you. Thank you.
All right. Thank you once again. A million thanks, Dr. Rose Ignatius, for your very informative, insightful, and inspiring talk this evening. You have enlightened all of us, and you see the number of questions from the participants. And that's actually the beauty of the science forum also. And great having you with us.